Before beginning the study of blood, we'll look at both the functions of blood as well as diseases of the blood. Blood has many functions. The first function we'll look at is transport. Blood transports respiratory gases O2 and CO2 between the lungs and the tissues. It transports nutrients between the GI tract, the gastrointestinal tract, and all other tissues. It gets rid of metabolic wastes. It moves hormones around the body, and it dissipates heat. Another function of blood is regulation. Blood regulates a number of different things, including your body temperature, a normal pH, osmolarity, and your electrolyte balance. Your blood protects you against infection and against its own loss. We're going to take some time to look at the composition of blood. Blood has two major components. There are formed elements, which are the actual blood cells, and there's plasma, which is a very complex aqueous solution. Let's look first at the formed elements. Formed elements are cells, cell remnants, and cell fragments in the blood. There are three major types: erythrocytes, RBCs or red blood cells, leukocytes, WBCs or white blood cells, and thrombocytes, also known as platelets. All three formed elements come from the hemocytoblast, and all are made in either red marrow or in lymphoid tissue. Erythrocytes, or red blood cells, make up about 95% of the formed elements in our blood. They are the simplest cells of the body because when they're mature, they have no organelles, including a nucleus. They are biconcave discs, so they look sort of like a lifesaver, but have a very thin middle. Erythrocytes are about 1/3 made up of a molecule known as hemoglobin. The main function of hemoglobin is to transport oxygen. A secondary function of hemoglobin is to transport carbon dioxide out of the body. Hemoglobin is a red protein that's responsible for transporting oxygen in the blood. Its structure is based on a theme of 4. It contains 4 polypeptide chains, 4 heme groups, 4 iron atoms in the center of each heme group, and each hemoglobin molecule has the ability to carry 4 molecules of oxygen. Take a look at the structure on the right side of the slide. Red blood cell production is also known as erythropoiesis, and it takes place in the bone marrow under the control of a hormone known as erythropoietin. We'll talk more about erythropoietin later. Red blood cells live about 120 days or 700 miles, and when they wear out, old cells are removed by the spleen or the liver. At first glance, this slide may look overwhelming, but let's go through it step by step. In the last slide, I mentioned that old red blood cells are removed from circulation by the liver and spleen. What happens to it after it goes to the liver or spleen? The iron is removed and recycled, the amino acids are recycled, and the heme is degraded. Bilirubin is produced, and bilirubin moves on into the blood. Bilirubin is a yellow pigment. The liver removes bilirubin and produces bile. So what happens if bilirubin builds up in the blood? Remember that bilirubin was the yellow pigment. The result is jaundice, also known as hyperbilirubinemia. Hyper means too much. Bilirubin refers to the yellow pigment bilirubin, and emia, the blood. Jaundice is a yellow discoloration of the skin, the mucous membranes, and of the whites of the eyes due to elevated levels of the chemical bilirubin in the blood. Jaundice is a disease condition characterized by yellow skin and the yellow sclera of the eyes. That's the part of the eye that's normally white. It's due to the accumulation of bilirubin in these tissues. Although bilirubin can accumulate other places, these are the two most common. It's a result of hyperbilirubinemia. Again, hyper means more, bilirubin refers to the bilirubin pigment, and emia refers to the blood. Hyperbilirubinemia is the buildup of bilirubin in the blood. There are 3 main reasons for jaundice or hyperbilirubinemia. They are the abnormal rapid destruction of red blood cells, liver disease or liver immaturity like you may see with a preemie, and blockage of the biliary system. To assess a newborn for jaundice, most often the physician will do a heel stick. TSB testing, or total serum bilirubin testing, is the standard for bilirubin measurement, which is measured directly from a blood sample and has to be obtained with a heel stick. TCB screening is non-invasive and measures bilirubin levels in the skin to provide an estimate of the bilirubin level. This picture shows you the TCB measurement being taken by a jaundice meter. The most common treatment for jaundice is phototherapy. The blue light helps reduce high levels of serum bilirubin. According to the clinician's decision, a blood transfusion is used when the bilirubin level is at a high level. Erythropoiesis means the production of new red blood cells. It is controlled by a hormone known as erythropoietin, which is produced by the kidneys. The effect of this hormone is to increase red blood cell production. Here's a short video to introduce you to erythropoiesis. What factors trigger an increase in the process of erythropoiesis or red blood cell production? They are blood loss, exposure to high altitudes, and red blood cell destruction. The slide on the right shows you the normal concentration of red blood cells. There are a number of different types of erythrocyte problems. The first we're going to look at is anemia, which means too few red blood cells. An means without, and emia refers to the blood. The first type of anemia is called hemorrhagic, and that's due to blood loss. Hemo again refers to the blood, and rhagia or RRHAGIA, the suffix means an abnormal or excessive flow or discharge. Hemolytic is the abnormal destruction of red blood cells, and again, hemo is the blood, lytic or lysis is to break apart. Aplastic anemia, inhibition of red marrow. Iron deficiency anemia, which means the inadequate dietary intake of iron. Protein deficiency is the inadequate dietary protein intake. Pernicious anemia means a vitamin B deficiency, and sickle cell anemia is a hereditary disorder. Take a look at these two blood slides. The one on the left is a normal blood smear. The one on the right is anemia. There are fewer cells in the one on the right, and also many of the red blood cells are misshapen. Another type of erythrocyte problem is polycythemia. Poly means many, cyth means cell, and emia refers to the blood, so there are too many red blood cells. There are two kinds of polycythemia. Secondary polycythemia, which can be a normal reaction to chronic exposure to high altitudes, or it can be caused by disease. And we're talking about things like emphysema or COPD, which is chronic obstructive pulmonary disease. Polycythemia vera is true polycythemia and usually caused by bone marrow cancer. The red blood cell count can get extremely high and the blood becomes very viscous. The blood in some cases becomes so thick that it's very difficult to pump. Here are two blood slides. The one on the left is normal. The one on the right shows polycythemia. Do you see how many red blood cells there are in comparison to the normal smear? In a minute, we're going to talk about hematocrits, and I'm going to show you the difference between a normal hematocrit and the hematocrit of someone with polycythemia. So how do you know if you have the right number of formed elements, which are primarily red blood cells? The answer is you do a hematocrit. A hematocrit tells you what percentage of your blood is actually red blood cells. So if you have a value of 25%, it means that there are 25 milliliters of red blood cells in 100 milliliters of blood. So how is a hematocrit done? Often by a manual method using a centrifuge. The first step is to collect blood in a centrifuge tube and seal the end. When collecting a blood sample using a centrifuge tube, it's important to use heparinized tubes so the blood does not clot. It is important to seal the end of the tube with Critis seal so that the blood does not come out in the centrifuge. The next step is to spin the blood sample in a centrifuge. During this process, the heavier formed elements, like red blood cells, will move to the outside end of the tube. In this centrifuge tube, you can see the heavier formed elements on the right side near the Critis seal, and on the left side you can see the plasma or blood serum. The next step is to read the hematocrit, and this is done using a hematocrit card reader as seen in the picture. To determine a reading, place the hematocrit tube on the chart. The The end of the tube with the critosseal should touch the bottom red line. The bottom of the blood column should touch the 0% line. Now slide the tube along the chart until the top of the blood column reaches the 100% line. Now read the top of the red column on the scale to the right. Remember the things we said to do on the last slide when reading a hematocrit tube. Take a look at this picture and see if you can determine why the value is 47%. What is the value of this hematocrit? The answer is on the following slide. The answer to the previous hematocrit value was 50%. In this slide, you can see two different hematocrits. The one on the left is a normal hematocrit. The one on the right is the hematocrit of someone with polycythemia. Remember, polycythemia means too many red blood cells, so it would make sense that their hematocrit would be higher. This is a labeled hematocrit tube. At the bottom is the critosseal. Just above that are the packed red blood cells that you see after centrifuging. In the middle of the packed red blood cells and the plasma is a very thin layer known as the buffy coat, and at the top is the plasma or serum. If you go back and look at the hematocrit tube in the previous slide, you would see that about 54% of this is blood plasma. About 45% of this blood is red blood cells, and about 1% of the blood is white blood cells and platelets, which make up the buffy coat. The average hematocrit value is 45%, but there's a difference on the average in males and females. Females usually have hematocrits that range from 36 to 40%, and males' hematocrits range from 40 to 50%. Let's look next at the leukocytes, or the white blood cells, which are larger and more complex than red blood cells. They are also fewer in number than red blood cells. There are two major categories of leukocytes. They are the granular and agranular leukocytes. The granular leukocytes are produced in red bone marrow. They have lobed nuclei and cytoplasmic granules, and there are three types of granular leukocytes: neutrophils, eosinophils, and basophils. The first granular leukocyte is the neutrophil, and you can see it pictured in the center of this slide. It has granules that stain light pink and light purple, and the one that we're looking at here is a segmented neutrophil, also called a polymorphonuclear leukocyte. It's the most abundant type of white blood cell in humans. Another granular leukocyte is the eosinophil, named this because it picks up the dye eosin, which is a red dye, in its granules. It's a normal type of white blood cell that has very coarse granules in the cytoplasm. Remember that the neutrophil, eosinophil, and basophil are all called granular leukocytes because they have granular type substances in their cytoplasm. A basophil is a type of immune cell that has granules with enzymes that are released during infections, allergic reactions, and asthma. A means without, so agranular means without grains. Agranular leukocytes have no granules in their cytoplasm. They develop from lymphoid tissues, and their nucleus is not lobed. There are two types, lymphocytes and monocytes, and we'll look at each. The first agranular leukocyte is a lymphocyte. It's a type of white blood cell that's part of the immune system, and because it's agranular, there are no grains in its cytoplasm. There are two main kinds of lymphocytes. They are B cells and T cells. The B cells produce antibodies that are used to attack invading bacteria, viruses, and toxins. The T cell plays an important role in immunity. The largest white blood cell is the monocyte. The job of the monocyte is to fight certain infections and to help other white blood cells remove damaged tissues or dead cells, and destroy cancer cells and regulate immunity against foreign substances. Leukocytes, or white blood cells, have many functions, but their main function is defense. Here are the 5 leukocytes and the role that each plays in the defense of the body. 2 of them are very important phagocytes, or phagocytic cells. A phagocyte is a cell that protects the body by ingesting, also known as phagocytizing, harmful foreign particles like bacteria and dead or dying cells. Those 2 phagocytes are neutrophils, which are the most abundant, and monocytes that have the ability to wander from the blood into the injured tissues. A less important phagocyte is the eosinophil. They phagocytize antibodies and release anti-inflammatory chemicals. Basophils release histamines, which promote inflammation, and lymphocytes produce antibodies and attack infecting agents directly. I mentioned the term inflammation. It's important to know the 5 primary signs of inflammation. They are rubor, redness, calor, heat, dolor, pain, tumor, swelling, and funkiolacea, which is loss of function. Be sure you know these signs. As you look at this picture, you can see 2 of the 5 signs of inflammation. You can see the rubor or redness and the tumor or swelling. What you cannot see is the heat, calor, the pain, dolor, and the loss of function, funkiolacea, although to look at this picture, all of those likely exist as well. To the right is a picture of another blood cell, or actually a blood cell fragment, known as a platelet or a thrombocyte. The most important function of thrombocytes is to stop bleeding. They are, in fact, cell fragments. They split in the bone marrow. They have a lifespan of about 5 to 9 days. They stimulate the formation of blood clots and sometimes a condition known as DVT, deep vein thrombosis, and we'll look at that on the next slide. Deep vein thrombosis, or DVT as it's abbreviated, occurs when a blood clot, also known as a thrombus, forms in one or more of the deep veins in your body, usually in your legs. Deep vein thrombosis can cause leg pain or leg swelling, but sometimes it occurs with no symptoms at all.